changes in the magnitude of quadratic expression, range of expressions and related problems. The rest of the problems in exercise 3 various quadratic equations and methods to solve them.
Good morning, students. This is Rama JL in Botany, and welcome to webinar 5 session. Today, we are going to discuss the chapter Photosynthesis. In that, C4 pathway. Yesterday in our class, we discussed about the photorespiration, which is a problem for the plants to face it. And to avoid that photorespiration, plant shows some adaptations. And in those adaptations, one of the adaptations is C4 pathway. And this pathway was discovered by Hatch-Slack scientists. Hatch-Slack scientists, and by the name of those scientists, this cycle was called as C4 pathway or Hatch-Slack cycle, and they gave the complete changes during this cycle. So C4 pathway is also be called as Hatch-Slack cycle. Now, which plants are included under this C4 plants? That means plants with those who exhibits the C4 characters or C4 adaptations are called as C4 plants. For example, maize, sorghum, sugarcane, and euphorbia. All these plants, they comes under C4 plants, or they show some C4 adaptations. And this C4 plants, which shows adaptations to avoid photorespiration. And what are those adaptations? How they plants adapt themselves. And if we observe the C4 cycle very in detail, by that we can understand this C4 cycle is actually a cycle which is evolved to avoid photorespiratory loss. So, in our previous chapters, we discussed about the photorespiration and its problems. And this main photorespiration problem is because of the changing behavior of rubisco. Yesterday in our class, we discussed it in a very detailed manner. What is the problem of rubisco? Means its nature changes according to the conditions. According to the conditions like high oxygen concentration and high temperature. These two conditions, they affects the nature of rubisco. And according to that conditions, rubisco originally it acts as a carboxylase enzyme, but when the conditions are with a high oxygen concentration and a high temperature, this rubisco that acts as oxygenase instead of carboxylase, it acts as oxygenase, and that becomes a hurdle for the normal plants. That's why to avoid that situation, whenever that comes, this rubisco, if it changes its nature, then plants, what should they do? That means by showing some adaptations, these plants, that means like maize, sorghum, sugarcane, like that, those plants, they show some adaptations, show some changes in their anatomy. And by showing that anatomical changes, plants, they escapes from the photorespiration. Then until and unless the plant shows some adaptations, they suffers from extreme conditions like high temperature and high oxygen concentration. So this is about the C4 pathway introduction. Why C4 pathway is? Means C4 pathway is mainly to escape from the photorespiration. Why means? Because during photorespiration, the main important enzyme is Rubisco, and this Rubisco do not show a flexible or do not show a fixed character. That means according to the situations like a high oxygen concentration and high temperature, those conditions influences the character of Rubisco. So at that time, this Rubisco that acts as oxygenase, and these conditions are this Rubisco oxygenase that is not suitable for this normal growing plants. So the plants definitely who are under compulsory conditions of this high temperature and high oxygen concentration should have some adaptations to escape from the situations. So that is the importance of C4. Then so here yes. Stuck అయింది. ఇది నెక్స్ట్ స్లైడ్ ఒకసారి. Then let us discuss about the reasons for adaptations. C4 plants, they are showing some adaptations. And what are those adaptations and how they helps in avoiding this photorespiration? Okay. There are two important adaptations in C4 plants. Those are number one, dimorphic chloroplast, and the other is Cringe type of anatomy in leaf. So these are the two important adaptations in C4 plants. Let us discuss about the dimorphic chloroplast. What is dimorphic? Di means two, morphic means morphologically, and morphologically they shows two types of chloroplast, and those are, one is Granule chloroplast and the second one is Granule chloroplast. So dimorphic means two types of chloroplasts are visible in this C4 plants, and those chloroplasts are number one chloroplast, Granule type of chloroplast. The other one is Granule type of chloroplast. Now let us see the Granule type of chloroplast. What is Granule? What is Grana, and how these chloroplasts are called as Granule type of chloroplasts? Before that, we should have idea about the structure of chloroplast. Yes, you know the structure of chloroplast. Just recall it. Yes. See here, this is a structure of chloroplast in which you'll find two membranes. That means each chloroplast is bounded by two membranes. That they are the outer membrane and the second one is inner membrane. Between these two membranes, there is some gap, which is called as intermembrane space. And next to that is inside the inner membrane, you'll find a gelatinous substance which is called as stroma. That is called as stroma. And in this stroma, you'll find so many groups of structures. So many groups of structures are visible in this stroma, and each group is called as grana. Then what is grana? Grana is say a group of this grana. In this, you are able to see plate-like structures, and these plate-like structures are arranged in one another. See this group also. Here, the number is not fixed. This plate-like structure that differs in the number. So, plate-like structures, they are inserted in one another. As a result of this inserting, these groups of structures are developed, and each group is called as a grana. Each group is called as grana, and this plate-like structure is treated as thylakoid. So the main important structure is thylakoid, and which is a plate-like structure, and these thylakoids, they are inserted in one another. As a result of this arrangement, groups of structures are developed, and each group is called as grana, and in each grana there are so many plate-like structures which are called as thylakoid. And in each thylakoid, pigment material, color giving material, that is called as pigment, and these pigments are stored in this thylakoid. Okay, if these are chloroplasts, then this the preserves chlorophyll A, chlorophyll B, like the pigments, those are preserved in this thylakoids. So, and these groups, they are connected with one another by means of, see here, some filament-like structures, and these are called as stroma lamellae. See here, lamellae. These are called as Fred channels or stroma lamellae. So this is the normal structure of a chloroplast. It was discussed in our previous class only. Then now come to the yes. Now come to this point. Here, a grana type of chloroplast. That means just now only discussed about the grana. Grana, what is grana? Is a group of thylakoids. Group of thylakoids are called as grana. And chloroplasts which have granas in their structures are called as granule type of chloroplast. Look at this diagram. Grana type of chloroplast. Yes. See here. Here, MCP, that means Mesopic Chloroplasts. In mesophyll tissue, chloroplasts are found, and which belongs to grana type of chloroplast. So here we are looking some dark spaces, which indicates this grana. This is one chloroplast in which these dark places are the indicators of a grana, and these granule groups are connected by light colored structures which are called as lamellae. That means in grana type of a chloroplast structure is similar to that of normal chloroplast. That means granule groups are arranged here and there in the chloroplast, and we know the granules are developed by the accumulation of thylakoids or arrangement of thylakoids in one another. So here these granules are connected with the light color structures which are called as stroma lamellae or thread channels. So it is similar to that of chloroplast structure. So there is no difference between chloroplast structure and a grana type of chloroplast, isn't it? Right. So in grana type of chloroplast, yes, in grana type of chloroplast which would have grana in the chloroplast are called as grana type of chloroplast. Then typical chloroplast and thylakoids would be arranged in stacks called as grana. Connecting to grana would be stroma lamellae. Then thylakoids have pigments which are the sites for non-cyclic photophosphorylation. We know the thylakoids. Thylakoids, they uh, thylakoids they help in storing the pigments, and pigments are wherever the thylakoids helps in storing the pigments, which are the sites for non-cyclic photophosphorylation. And grana chloroplasts, they are usually found in mesophyll cells. They are found in mesophyll cells. Right. So, granule type of chloroplast, they resembles the structure of normal chloroplast. Structure will be the same, but they are found in mesophyll cells. They are abundantly found in mesophyll cells of the leaf. That's it about the granule type of chloroplast. Then the second one is agranule type of chloroplast. Agranule. A means absent. What is absent? Grana. Grana is absent. Then what are the remaining structures? Means without grana means only thread channels or stroma lamellae are present in this type of chloroplast. So this type of chloroplast would not have grana. They would not have grana. That means grana is absent, or granas are absent in this type, and they would have only stroma lamellae. And I'll show you the concerned diagram. Let's see here. In this type, a BSCP, that means bundle sheath chloroplasts. This type of agranule type of chloroplasts are found in bundle sheath cells. You know the bundle sheath. What is bundle sheath? Yes, it is a layer. Yes, here it is bundle sheath. This bundle sheath is a layer which helps in surrounding the vascular tissue. See here, xylem and the phloem tissues are there. This is xylem and the blue color is phloem tissues. And this vascular tissue, both together are called as a vascular tissue. And this vascular tissue is surrounded by a single cell thickness, one cell thickness layer, and this layer is called as bundle sheath. So in this bundle sheath cells, you'll find this agranule type of chloroplasts. Agranule type of chloroplasts are visible, or they are found in bundle sheath cells. of a leaf. So, a granule chloroplasts are found in the bundle-shaped cells. Okay, there's are two types of adaptations are found in dimorphic chloroplasts. So, in C4 plants, to avoid this photorespiration, the plants should have this adaptation. Why means to avoid the photorespiration. By having this granule type of chloroplast and a granule type of chloroplast plants, they are able to escape from the photorespiration. How means that will be discussed in further slides. Then next, second type of adaptation. What is that? That is Cringe type of anatomy. What is Cringe type of anatomy? That means in C4 plants, Cringe type of anatomy means the cells of bundle sheath are arranged in a circular shape. Just I want to be observed it in the diagram once again. Yes, see here, this bundle sheath cells, they are arranged in a circle, in a round structure, in round shape. And cells, they are one cell thickness. They are only one cell thickness, and these cells are arranged one after the other. So this type of arrangement is also called as a reed-like manner. Reed-like means in a circular manner, the cells are arranged in this bundle sheet, and this type of adaptation is also helpful in avoiding the photorespiration, and such type is called as Cringe type of anatomy. So this Cringe type of anatomy, yes, here also you find this Cringe type of anatomy. See here, the centrally located tissues are vascular tissues which are surrounded by a large big cells, and this single cell thickness, one after the other cells arranged layer is called as bundle sheath. The cells of bundle sheath with the special type of chloroplast, and this chloroplast, they belongs to the a granule type. So granule type of chloroplasts are found in mesophyll cells, and a granule type of chloroplasts are found in vascular bundle cells. So this Cringe type of anatomy and a dimorphic chloroplast, both are the adaptations shown by C4 plants in escaping the photorespiration. That's the next. These are the adaptations. Right. So, in adaptation, the main important points are dimorphic chloroplasts. In dimorphic, again, granule type of chloroplasts are there, which are found in mesophyll cells, and these granule type, they resemble the structure of chloroplast. And the second one is a granule type of chloroplast, and which are found in bundle sheath cells. And this a granule type of a chloroplast, they lack the grana stacks. And the second adaptation is Cringe type of anatomy, in which the cells are arranged in a wreath-like manner. That means cells are arranged in a circular manner, and these two are the adaptations shown by C4 plants. Yes. Now, mechanism of C4 cycle. This C4 cycle involves two different parts of the leaf. Those are number one, upper mesophyll cells, and the second one is bundle sheath cells. In these two parts, in these two different parts, the cycle will be completed. So two types of cells are involved in completing this cycle. The first one is upper mesophyll cells and the second one is bundle sheath cells. Okay, in first, what is that mesophyll? Yes, in mesophyll cells, how the C4 pathway takes or that undergoes the changes. Let us, C4 cycle starts by absorbing the CO2 from the upper surface of the cells, and that means CO2 diffuses into the mesophyll cells and it reacts with the H2O in presence of enzyme carbonic anhydrase. That is carbonic anhydrase, not carbonic acid, carbonic anhydrase enzyme that helps in reacting CO2 with H2O. There is a faster change during the C4 cycle in mesophyll cells. So, the faster change of this C4 cycle is uh, absorption of carbon dioxide from the from the atmosphere into the mesophyll cells, and that carbon dioxide reacts with the H2O molecule, water molecule, in the presence of one enzyme which is called as carbonic anhydrase, and this reaction results in the formation of what um, and it results in the formation of carbonic acid. So, under the influence of a carbonic anhydrase enzyme, CO2 reacts with the H2O and results in the formation of carbonic acid. And this carbonic acid in mesophyll cells only, it dissociates into H plus and the bicarbonate. Now, this bicarbonate, it is accepted by PEP. What is PEP? Phosphoenol pyruvic acid, which is a 3C compound. It is a 3C compound, 3-carbon compound. So, here what we have to understand is in C3 cycle, CO2 is directly it is accepted by RUBP in the presence of Rubisco. But here CO2 is not directly accepted by PEP, isn't it? So CO2 is not this is not combining with the PEP directly. First, the CO2 is reacting with the water molecule in the presence of a carbonic anhydrase enzyme and helps in the synthesis of carbonic acid. And now this carbonic acid is dissociating into proton and bicarbonate. Then this bicarbonate is accepted by PEP. That means CO2 is not directly accepted by PEP. In the form of bicarbonate, it is accepted by PEP. What is PEP? Phosphoenol pyruvic acid, which is a 3C compound. And by the acceptance of this bicarbonate by PEP, it combines with the bicarbonate and forms a 4C compound, which is called as oxaloacetic acid. That is OAA, oxaloacetic acid. A 4C compound is synthesized by the combination of PEP with the bicarbonate. PEP with bicarbonate. Now, here this 4C compound, it is the first product of this cycle. That's why this cycle is called as C4 cycle. Why it is called as C4 cycle? Means the first formed product is a 4C compound. The first formed, first synthesized compound is a 4C compound. That's why it is called as C4 cycle. And this is a carboxylation reaction. Yes, we know that because CO2, acceptance of CO2 by and this PEP is a carboxylation reaction. And for this carboxylation, it requires enzyme. Definitely, it requires one enzyme which is called as PEPCO. PEP means phosphoenol pyruvic acid. Co means carboxylase. Why? Because this is a carboxylation reaction. Acceptance of carbon dioxide in the form of bicarbonate by PEP. that is taking place during in this mesophyll cells. So, a resultant first synthesized or first formed product is the 4C compound. That's why it is called as C4 cycle. And, during this carboxylation, one enzyme is required, and that enzyme is PEPCO. PEPCO means phosphoenol pyruvic carboxylase. Under the influence of this carboxylase enzyme, this OAA is synthesized, which is a 4C compound. Now, what is the next step? Third step. First step is entry of CO2. See here, first reaction in the or first change in the mesophyll cells is, once again I am repeating, in mesophyll cells CO2 which is absorbed from the atmosphere through the upper surface of the cells, and that diffuses into the mesophyll cells. There it reacts with the H2O molecule by the presence of enzyme carbonic anhydrase and gives carbonic acid. This is the first step. In second step, this carbonic acid dissociates into proton and bicarbonate, and this bicarbonate, it combines or it is accepted by phosphoenol pyruvic acid, which is a 3C compound, and by the combination of these two, a 4C compound is produced, which is called as oxaloacetic acid. This is the second. That means addition of a bicarbonate with the BEP results in the formation of 4C compound like oxaloacetic acid. And because of the formation of 4C compound, it is called as C4 cycle. And this oxaloacetic acid a synthesis is a carboxylation reaction where enzyme is required, and that required enzyme is PEPCO, phosphoenol pyruvic carboxylase. That is the enzyme, and under the influence of this enzyme, under the influence of PEPCO, OAA is synthesized. Now, next change in the mesophyll. What does that means? This OAA that can change into two products. One is malic acid, or by adding with, that means OAA, when it combines with one amino group, it results in the synthesis of aspartic acid. So there are two ways. That means this OAA can change into malic acid or aspartic acid. Malic acid is directly synthesized, but this aspartic acid, it is synthesized or it is produced when this OAA is added with amino group, then only aspartic acid is synthesized. So whatever may be the end product of this OAA conversion, they enters or they diffuses into the bundle sheet cells. That means if this OAA if it changes into malic acid, then it diffuses into the bundle sheet cells only, or else if OAA by adding with amino group, if it changes into or if it becomes as aspartic acid, then this also diffuses into the bundle sheets. So whatever is the end product of OAA conversion, they diffuses into the bundle sheet cells. So these are the important steps in C4 cycle which takes place in mesophyll cells. So these are the important changes. Then next, look at the changes in bundle sheet. Yes, this is the leaf anatomy. Just now what we discussed is seen here. So these are the upper epidermal cells, and here and there stomata are arranged, and these light color cells are mesophyll cells and palisade cells, and these are spongy cells. And so here from this upper epidermis are two stomata. CO2 whatever is present in the atmosphere that diffuses into the mesophyll cells. There the changes starts. That CO2 combines with the H2O which is present in the cells of the mesophyll, and by the influence of carbonic anhydrase it becomes as carbonic acid, and carbonic acid it dissociates into protons and bicarbonate. Then the next changes that undergoes, and finally the end products of this mesophyll changes in mesophyll are malic acid or aspartic acid. This malic acid or aspartic acid, which is formed in these mesophylls, diffuses into the bundle sheath cells. With that, mesophyll part completed. Then next, come into the bundle sheath. Yes. C4 pathway in bundle sheath cells. C4 changes starts by the diffusion of malic or aspartic acid into the bundle sheath. Yes, it starts, that means bundle sheath C4 cycle in bundle sheath cells starts by the diffusion of malic acid or aspartic acid. Then, what is the importance of these two means? Malic acid or aspartic acid, both are 4C compounds. Malic acid is a 4C compound and aspartic acid also a 4C compound. So, when malic acid, for example, if malic acid diffuses into the bundle sheath cells, thinking, then what happens in the further changes? That means malic acid splits into two compounds in the bundle sheath cells. One is pyruvic acid, which is a 3C compound, and one carbon dioxide molecule, in which one carbon molecule is there. So, malic acid, as we are saying that malic acid is a 4C compound. So, that means it holds 4 carbons in its structure. So, this malic acid is splitting into two different compounds. One is 3C compound, the other one is 1C compound. And this 3C compound is called as pyruvic acid, and the 1C is carbon dioxide. Now, this carbon dioxide, whatever is synthesized in the bundle sheath cells, that will be released in CO2, which is released in the bundle sheath cells, that combines with RUBP for the running of a C3 cycle in bundle sheath. That means here in bundle sheath cells, whatever carbon dioxide is produced is helpful in continuing the C3 cycle. And what about the pyruvic acid? What does the role of this pyruvic acid? Yes, pyruvic acid that diffuses into again mesophyll cells. That diffuses into the mesophyll cells. That means that comes out of the bundle sheath cell and enters into the mesophyll cells. And it is in its structure, it is without phosphate group. In pyruvic acid structure, there are no phosphate groups. That's why definitely it undergoes phosphorylation. We know the meaning of phosphorylation. That means utilization of ATP. Yes, here one ATP molecule is required for this phosphorylation. And here we have to understand one thing. In ATP, there are on the abbreviation of ATP, adenosine triphosphate. That means tri means 3, phosphates means phosphate groups. So there are 3 phosphate groups. There are 3 phosphate groups, and out of this 3, When the ATP splits or when the ATP is utilized during this phosphorylation, that means it splits into three uh, inorganic phosphate radicals. And out of those three, two inorganic phosphates are utilized during this phosphorylation. That means the end product of a phosphorylation from ATP is AMP. That means adenosine monophosphate. That means this adenosine that holds only one inorganic phosphate, and by relieving two inorganic phosphates for this this indicates the utilization of two ATP molecules during this reaction or during this change of pyruvic acid into PEP. What is PEP? We know it already that PEP means phosphoenol pyruvic acid. So for the regeneration of PEP, we are saying regeneration. That means initially we have discussed in this, just I'll show you. Yes, in mesophyll, bicarbonate is combining with or bicarbonate is accepted by PEP. That means from where this PEP is coming or from where it is coming. That means this PEP is produced in bundle shape cells. Yes, see here only. In bundle sheet changes, we are observing the changes in bundle sheet. That means when this malic acid is diffusing into the bundle sheet cells, that malic acid is dissociated into two compounds. One is 3C compound and the other one is 1C compound. And this 3C compound is pyruvic acid. Pyruvic acid that diffuses into the mesophyll. Before diffusing into the mesophyll cells, it undergoes phosphorylation by utilizing one ATP molecule. Here, if we observe it in deeply, this ATP molecule is having three inorganic phosphate radicals. Out of three, two inorganic phosphate radicals are utilized during the phosphorylation. So, leftover inorganic phosphate molecules are only one. That means the end product is AMP. That means ATP changes into AMP during the phosphorylation. So, two inorganic phosphate radicals are utilized by the pyruvic acid and that turns into phosphoenol pyruvic acid. Then it diffuses into the mesophyll cell and that again helps in regenerating again oxaloacetic acid and the malic acid like that. So with this, the main important change in the bundle sheet is over. The next, what about the carbon dioxide? What is the role of carbon dioxide which is released in bundle sheet cells? If we discuss it about this carbon dioxide, this carbon dioxide which is released in bundle sheet cells combines with the RUBP. We know RUBP is a CO2 acceptor and this RUBP by absorbing or by accepting the CO2 molecule that continues the C3 cycle in bundle sheet cells. And the final end product of this C3 cycle is glucose. Glucose will be synthesized in bundle sheet cells. And for this, it requires enzyme like Rubisco. What is Rubisco or UBP? Carboxylase. Why? Because here CO2 is accepted by RUBP. Carboxylation reaction is taking place in bundle sheet cells. That's why here Rubisco acts as carboxylase. Now, the main intention of C4 pathway. Here, what are the adaptations? It shows during this C4 pathway. Means, here the upper epidermal cells. Here, this upper epidermal cells, they are diffusing temperature and carbon dioxide into the mesophyll cells. And As a result of this very closeness to the upper epidermal cells, in mesophyll cells, the temperature is more than it is more than that of bundle sheet cells. That means in the inner parts, temperature is less, whereas the tissue which is very closer to the upper epidermal cells are having more temperature. And next, one more thing, with this mesophyll cells, they are having Grana type of chloroplasts. So where there is Grana type of chloroplasts, they are having thylakoids. So this thylakoids, by having pigments, they performs light reaction in which cyclic and non-cyclics are there, and during cyclic or non-cyclic, assimilatory power will be synthesized, and then by utilizing this along with assimilatory power, oxygen is also be released during non-cyclic photophosphorylation. So here concentration of oxygen gradually it increases, and temperature it is more than the inner tissues, and these two conditions do not support the C3 cycle or enzyme Rubisco, which Rubisco is essential to perform C3 cycle, but C3 cycle is influenced by RUBP carboxylase, and RUBP carboxylase is not active under this high temperature and high oxygen concentration. So this is a hurdle for the plant. So to avoid these conditions, that means under this high temperature and high oxygen concentration, RUBP acts as an oxygenase, and RUBP oxygenase cannot move the C3 cycle. That's why the C4 plants, they shift C3 cycle into the bundle sheath cells where temperature is less than the mesophyll cells, and the oxygen concentration is also less than the mesophyll cells. That's why in bundle sheath cells, this C3 cycle will be performed by the bundle sheath cells. And here, advantage of this plants is shifting of C3 cycle into the bundle sheath cells is one more advantage. Why? Because in bundle sheath cells, temperature is less and the oxygen concentration is also be very less. Under these conditions, Rubisco acts as carboxylase. So under the influence of this carboxylase, Rubisco carboxylase, the CO2 will be accepted by RUBP and it continues the C3 cycle, in which the starch will be the end product of the process. Right. So in this way, total, what is that? C4 cycle will be completed. Then what about the pyruvic acid? And just now only we discussed it also. Then pyruvic acid undergoes phosphorylation by utilizing two inorganic phosphate radicals. See here, yes. Yes. So, by utilizing two inorganic phosphates, it becomes as PEP, and again that diffuses into the mesophyll cells for the recycling of a C4 pathway. And here in C4 plants, C3 cycle is shifted into the bundle sheath cell, and here Rubisco acts as a carboxylase, and here for problem of photorespiration will not be seen in C4 plants. So in this way, C4 plants exempts they avoids the photorespiration and continues the C4 pathway by showing these adaptations. And here, one more topic. What is the advantage of this anatomy? That means we are saying that dimorphic chloroplast, Cringe type of anatomy, shifting of C3 from mesophyll into the bundle sheath cells. All these are anatomical advantages. Then what are the advantages of this anatomy? Means once again, granule chloroplast would be in the mesophyll cells and agranule chloroplast would be in the bundle sheath cells. Wherever there is granule chloroplast and thylakoids, they are there. There is a right site or right place to perform the light reaction. And wherever agranule chloroplasts are there, there is the stroma and the C3 cycle will be continued at that place. So anatomy, what is the anatomy means? The dimorphic chloroplasts, they are helping in performing what is a light reaction and also C3 cycle. That means a granule chloroplast, by showing dimorphic chloroplast, that means granule chloroplast, they are helpful in performing light reaction, and agranule, they are in the bundle sheaths. They are in the bundle sheath and helps in performing the C3 cycle. The next.
Welcome back to integrated second year English classes. I am Samantha.
Sure. So these are the advantages shown by C4 plants, and in order why they are showing these adaptations means to avoid the photorespiration and to continue the C4 cycle. With this, we'll stop it here, and tomorrow we'll see another topic.
Fine, sir. Fine. Class madam. లేదు మీరు రాలేదు ఇక్కడ కనిపిస్తలేదు